screen, man. Is it visible, ma'am?
Yeah, just put it on presentation mode.
Yeah, just. It is taking time actually. Is it in the presentation mode, ma'am?
Not yet.
I think I have to follow the procedure described by the Ravi Shankar sir yesterday.
Yeah, yeah.
Just a minute. I will share it. Stop share. But it is not showing, ma'am. Actually, after full screen, now I am unable to see the Zoom.
Able to see.
How to share now? Actually, screen is occupied fully. There is no other option.
Just try it again.
Just a minute. No, actually, I have not shared. I have just put it into this presentation mode. But after putting on the presentation mode, I am not getting the option for sharing. It means not no option for Zoom is sharing. It is showing.
Just give me 1 minute. I'll send Shekhawat and he'll help you. Okay.
I'm trying. Just a minute. इसके पास करना है ये कर दिया मैंने शेयर लेकिन ये हो नहीं रहा है कौन सी नहीं हुआ अब ये फुल स्क्रीन पे डाल रहा हूँ फुल स्क्रीन पे डालो हो नहीं रहा कंप्लीट वो तो नहीं वहां नहीं दिखा रहा हम मैडम को दिखा रहा मैम अभी फुल स्क्रीन पे शो कर रहा है नहीं सर अभी
No, it's still not there. Just give me a second. Shekhawat is there with you. He's coming to assist you.
ये भी फोन करना अब इसको करो
एक बार अगर नहीं दिखे ना तो दूसरी बार इसको शेयर कर दो
अच्छा
जो ऑलरेडी प्रेजेंटेशन मोड में
हाँ
अब दिखेगा
मैम इज इट इन द प्रेजेंटेशन मोड
It's visible now. Thank you, Shekhawat.
Very good morning to all of you. I am Dr. Jairam Chaudhary. I am going to present biofertilizers, their production, application and their significance into the agriculture. So friends, as we all know that at the time of independence, we were having only 50 million tons of the food grains, which were not sufficient to fulfill the demands of such a huge population. So during the 1960s, with the inputs like hybrid seeds, high yielding short duration crop varieties, better irrigation facilities and availability of the agrochemicals, we were able to achieve a quantum jump in the food grain production. But after that, farmers started use of these agrochemicals non-judiciously, which has created pollution and contamination of our soil and water. Like when we apply the chemical fertilizers, nitrogenous fertilizers, nitrate leaching to the groundwater can cause blue baby syndrome, which is also known as blue methemoglobinemia. And denitrification of these nitrogenous fertilizers contributes significantly to the greenhouse gases. These chemical pesticides residues are non-degradable. They have a long persistence period, so they can contaminate our food chain also, and time by time they bioaccumulate in our body, animal body, and in our ecosystem also. So these are hazardous to the environment, human and animal health. Application of these chemical fertilizers have also resulted in soil being deprived of essential plant nutrients and our organic matter content. As we know that when we apply NPK to the soil, microbes start flourishing, but they also need carbon, which they takes from the soil. We are not adding the carbon out from the extra inputs, so they will mineralize the soil organic carbon and it will be lost to atmosphere in the form of the CO2. So over the time span, there is a continuous reduction in the organic matter also. Then these agrochemicals harmfully affect our beneficial microorganisms also. So it ultimately reduces the soil fertility and makes our crops more prone to the diseases. It is just like that if a body part of human or animal is affected, it affects the whole body. The functioning is affected as a overall. So in the same way, we have also disturbed the microbial diversity or ecosystem of our soil. It was self-sustaining earlier, but now with the use of extra use of these synthetic fertilizers and pesticide, we have imbalanced it. So it has resulted into the emergence of new fertilizers and new pests and diseases. There are other constraints are also emerging, like yield stagnation in spite of the increased doses of the chemical fertilizers. Then low fertilizer use efficiency is also major constraint. So please mute yourself. So low fertilizer use efficiency. You know that when we apply the nitrogenous fertilizers to the soil, hardly 30 to 50% of our applied nitrogen is available to the plant. Rest of the nitrogen is either lost in the form of the leaching or through the volatilization. And this efficiency is much lower in case of phosphatic fertilizer, which is 20 to 30% only. That is because when we apply the phosphate to the soil, they form the complexes either with the organic matter or iron, calcium or zinc. So what happens if our soil is poor in microbial population or in diversity? So it will not be solubilized. But if our soil quality is good, we are having sufficient diversity of microbial agents which can solubilize the phosphorus. So whatever fertilizers we are applying, it will be available to the plant meantime, because by the several mechanisms, microorganisms makes unavailable phosphorus to the available form. So, and there will be an imbalance between nutrient mining and nutrient replenishment. We know that plant requires 17 elements, but as of today now, farmers are providing NPK. Some of the farmers are giving iron and zinc also, but it is very low percentage. We are not providing a complete set of nutrients to the soil. Complete set of nutrients to the soil can be provided only by means of the organic inputs. So if we do not follow that procedure, there will be a huge imbalance between the nutrient mining and nutrient replenishment. So our soil will become deprived of these essential nutrients over a period of time. So by this process, we are also increasing the burden on the our natural fossil fuel reserves, which are limited today. We are seeing that petroleum prices are increasing, crude oil prices are increasing, because we have limited fossil fuels reserves. So we have to conserve them also. So we have to minimize the application of chemical fertilizer, because these synthetic fertilizers are prepared by the use of these fossil fuels only. These fossil fuels play a role as energy source in their production. So what is the solution? How we can maintain our soil health and nutrient status so that we can achieve a sustainable production into the agriculture. So solution lies with the biofertilizers. Biofertilizers are the products containing living cells of the beneficial microorganisms that have an ability to mobilize the nutrients from non-usable to usable form through various biological processes like nitrogen fixation, phosphorus solubilization, K solubilization, zinc solubilization, iron solubilization, organic matter degradation. There are several biological processes in which they can benefit to the plants. Some of the famous microbial formulations are also available in the market now. So I have listed few of them here, like nitrogen fixers, which are broadly classified into the three categories: symbiotic, non-symbiotic, and associative symbiotic. Under the symbiotic category, we all know the Rhizobium, Frankia, Anabinase, cyanobacteria. So these are symbiotic. Symbiotic means they are physically attached to the plants. They make their own structure into the plant system, and then they fix the nitrogen. They depend on the plant for nutrient and energy, and they provide nitrogen to the plant. So it is a symbiotic association. Due to their symbiotic nature, they are host specific also. But coming to the non-symbiotic nitrogen fixers, they are Azotobacter, Beringia, Clostridium. There are many more examples are also there. They are free living in nature, so they can be applied to the wide range of the crops. But their efficiency is not as much symbiotic nitrogen fixers, which we will discuss later. Then there are associative symbiotic bacteria also, Azospirillum. It can also be used to the variety of crops. Then phosphate solubilizers. Phosphate solubilizers like Bacillus megaterium, Pseudomonas strata, Aspergillus, Penicillium, they solubilize the complexes of the phosphates, either organic phosphates or tricalcium phosphate, zinc phosphate, aluminum phosphate. By the various mechanisms, they break these complexes, either stop the formation of the complexes or break the complexes and make the phosphates available into the soil solution. Then there are phosphate mobilizers. Mobilizers means simply transporters. They transport the available form of the phosphate from deep soil horizons to the root zone. And the famous example is ectomycorrhiza, endomycorrhiza. Mycorrhiza play that role only. Fungal hyphae which penetrate deeper into the soil layers, they take phosphate from deeper horizons and transport to the root horizon. Then nowadays, potassium solubilizers, zinc solubilizers have also been discovered. Then some of the bacteria like Pseudomonas, Azotobacter, Azospirillum, they produce plant growth promoting hormones like auxin, cytokinin, gibberellin. So they support the plant's growth. Then in the disease control also, like Pseudomonas, Bacillus, Trichoderma, they have significant role in controlling the soil-borne diseases. Then Bacillus metarhizium, Beauveria bassiana, these are also used as a famous biocontrol agents. Then in the composting process also, we know that composting is totally dependent upon the microorganisms. But addition of some particular microorganisms like Aspergillus, Trichoderma, these are fast decomposers. So their addition to the compost pit can speed up the process of composting, and it may reduce the time of composting. And addition of the Azotobacter, Azospirillum during the curing phase, means after the decomposition, when our compost is decomposed and temperature comes down, at that time we can add some nitrogen fixers, phosphate solubilizers, so they improve the quality of our compost. Then there are several microorganisms which can decontaminate our degraded soils, like Pseudomonas, Xanthomonas, Thiobacillus, Rhodococcus, white rot fungi. They are efficient in degrading some xenobiotic compounds or organic organochlorines like our agropesticides. So they can be helpful in amelioration of our degraded soils. Then mycorrhiza have a very specific role in the plants. They prevent the plants from the drought stress. They cover the roots surface and prevent the root from desiccation during the drought time. So it helps to withstand the plant under the drought stress. Then there are several microbial pesticides also to control the insects, like Bacillus thuringiensis, most famous example, nuclear polyhedrosis viruses, Entomopthora is a fungi, then Trichoderma polysporum, then among the protozoan category Nosema lactusi. So then some of the organisms are also used as a microbial weedicide like Zygogramma bicolorata. One Parthenum rust has also been identified by our Dr. Chandrabhanu sir. He is doing experiment. That rust is also very effective in controlling the Parthenum disease, Parthenum weed. Then there are xenobiotic scavengers. Means I have discussed that amelioration of degraded soils. These Pseudomonas fluvibacterium, Rhodococcus nocardia, Micromonosporas proteasum, these bacteria are very efficient in degrading these xenobiotic compounds. Xenobiotic means the compounds which are hard to degrade, which are recalcitrant in nature. So they decompose those compounds are also. So some of famous biofertilizers we will discuss, like Rhizobium. It is symbiotic in nature and it is aerobic bacteria, but is highly host specific. It can fix nitrogen around 50 to 300 kg nitrogen per hectare per year. Media used for its isolation is Congo red yeast extract mannitol agar for those who want to start up a biofertilizer unit. So this is the media used for its isolation, and this is how Rhizobium looks like on the media plates. It looks from pinkish to the creamy white in color. Then Rhizobium is very host specific, and it is classified into the 7 cross inoculation groups. Cross inoculation groups like clover group, alfalfa group, bean group, lupine group, pea group, soybean group, cowpea group. Cross inoculation means a particular Rhizobium species like Rhizobium japonicum can inoculate only soybean group. It cannot be beneficial for pea group or lupine group. So they are very host specific to that particular group only. Coming to the Azotobacter, it is a free living bacteria. It can be useful for several crops. It can be used in the cereals, it can be used in the vegetables, it can be used in the fruit crops also. So, but its nitrogen fixation potential is very low as compared to the Rhizobium. It is 10 to 20 kg nitrogen per hectare per year, because nitrogen fixation is an anaerobic process. It requires an environment which is not having oxygen. So symbiotic bacteria prepare root nodules at the plant and then make the system anaerobic, so they do not want to, they will not waste their energy in the oxygen production. But these free living bacteria are present freely into the soil, so they have to avoid the oxygen stress also during the nitrogen fixation. So their efficiency is very low as compared to the symbiotic nitrogen fixers. So media used for its isolation is Jensen medium. These are available into the market. Then they are cyst forming, adverse resistant to the adverse environmental conditions. They also produces the growth hormones, which affect which is beneficial for the crop's growth. These are some famous species of Azotobacter: Azotobacter curcucum, Azotobacter vinelundai, Azotobacter pascali, Azotobacter salinestris, and Azotobacter pyrankite. Azospirillum is an associative symbiotic nitrogen fixer. Associative means it requires oxygen in a medium concentration. Means it is microaerophilic in nature, not aerobic, not anaerobic. It is microaerophilic. It shows subsurface growth. Those who want to check that whether I have isolated azospirillum or not, they can simply grow their culture into the media tubes, liquid media tubes. It shows subsurface growth. Aerobic bacteria shows the growth at the surface, but it shows the subsurface growth. There are the famous species like Azospirillum, Lycopharum, and Brasiliensis. Coming to the phosphate solubilizing microbes, how they solubilize the phosphates complexes like aluminum phosphate, iron phosphate, tricalcium phosphate, zinc phosphate. So few of mechanism I have listed here, like production of weak organic acids, phytases, phosphomonoesterases, or diesterases. These are the enzymes. Exopolysaccharide production, cytophore production, hydrogen ion extraction. You can see here simply, like if we see the phytases enzymes, these enzymes simply break the bond between organic compounds and phosphate and make the phosphate available to the plant. But if we look at the cytophore production, this cytophore chelates the iron molecule. So once they chelate the iron molecule, iron will not be available to the phosphate, so it will not form the complex and it will be available to the plant. By the production of EPS, exopolysaccharides, they can chelate calcium ions. So calcium ion will be trapped with exopolysaccharides, so phosphate will not form the complex with the calcium. Then with the acid production, acid will form the complex with the calcium and phosphate will remain into the soil solution, so plant can take up very easily. So in this way, microbes play in diverse manner. So they either prevent the complex formation or they break the complexes. So solubilize the phosphate and available to the plant. If we want to identify our bacteria whether it is solubilizing or not the phosphate, we can simply take the Picovaschia agar medium tube plates. We can put our culture onto the tube, is this plate, and after 5 to 6 days we can see a halo zone around the bacterial colony. This halo zone is only due to the phosphate solubilization. Rest of the area is not solubilized. Phosphate complex is present over here, that's why it is looking in white in color, but this halo zone is indicating the greater the halo zone, better is the capacity of microorganism to solubilize the phosphate. So this is the estimate of nitrogen fixation potential by microbial system. Like legume rhizobium system have a potential of fixing the nitrogen like 50 to 300 kg per hectare per annum. Cereals azoospermum system, it have 10 to 25 kg nitrogen per hectare per annum. Cereal cyanobacteria system, it have 10 to 30 kg nitrogen per hectare per annum. And rice azolla anabina, which is a very common famous example, it also has a potential of nitrogen fixation around 20 to 90 kg per hectare. So if we are applying these biofertilizers to our system, so with this calculation we can reduce our fertilizer dose and we can save that also that fertilizer consumption. These are the crops which can be benefited by these groups of bacteria. So for rhizobium, all are the leguminous crops. For azotobacter, we can see here that cotton, vegetables, mulberry plantation crops are also there, cereals are also there. Then sunflower, tobacco, fruits, spices, condiments. It has a wide range of crops which can be benefited by the azotobacter. In same way, azoospermum can also be used for the variety of the crops. Blue-green algae is mostly used in the high water requiring crops. Then azolla is specifically used in the rice, and phosphate solubilizing bacteria and fungi can be applied to the all crops because they are not crop specific. So coming to the biofertilizer, this was the benefits provided by the bacteria. Now coming to their production system. So how to produce the biofertilizers? So first of all, we will discuss about the characteristics of good inoculant strain. It should be prompt and effective in the nodulation, nitrogen fixation with the host cultivars under wide range of environmental conditions. Effective in nodulation, and it should be prompt, because if it is not prompt, then there are the bacteria into the soil which can form the nodules with the plant, which may not be efficient in fixing the nitrogen. So our desired bacteria should be very effective in the nodulation. They should have good survival onto the seed and fast multiplication into the soil so that they can compete with the native microflora. They should have greater enzyme production capability to solubilize the nutrients so that they can maintain their growth also. They should be competitive into the nodule formation. They should show good growth in culture and should have a good survival into the carrier so that we can store them for a longer period. A carrier material should also have good characteristics, like it should be non-toxic to the inoculant bacterial strain. It should have high organic matter content to support the bacterial life. It should be able to absorb the good amount of moisture. It should be easy to process and free of lump forming materials. It should be easy to sterilize by autoclaving or gamma irradiation. These are the methods used for their carrier matter sterilization. And it should be available in adequate amount throughout the year. It should be inexpensive, good adhesion to the seeds. Otherwise, if it is not having adhesion properties, how we will treat our seeds? Then it should provide the buffering capacity so that pH changes does not occur, and it should be non-toxic to plant also. There are two methods of carrier material sterilization. One is the gamma irradiation, which is most commonly used. It does not bring any changes to the physical and chemical properties of the material, so it is preferred. Autoclaving, when carrier material is packed and partially opened or thin walled polypropylene bags and then autoclaved for 60 minutes at 121 degrees centigrade and 15 psi lbs pressure. So it should be noted that during autoclaving, some materials changes their properties due to toxic substances. That's why gamma irradiation is most preferred method over the autoclaving also. Then there are steps. These are the steps in the inoculant or biofertilizer preparations. First of all, we have to isolate our desirable bacteria. Then we have to screen that bacteria for that particular function, whether it is having a greater efficiency or not. Then we have to maintain it. Then we have to multiply it. Then after multiplication, its incorporation into the carrier, then packaging, labeling. We can see through this flow chart. Like after the microorganism isolation, bacteria isolation, which is known as mother culture, we transfer into the new test tubes having the culture media, and then from that culture media we have to multiply it into the small flask. So we have to activate our culture. Then from small flask, a inoculum can be prepared to the larger flask. After that, we are having sufficient inoculum. We have to multiply into the larger volumes. So there are two methods nowadays. Automatic fermenters are available, which control every chemical and physical parameter like temperature, pH, oxygen concentration, nutrient requirement. Everything is automatically monitored in these systems. Shaking is also done by the help of its turbulence. Then another method is there if we are having shakers, incubator shakers. So we can use large size flask, and in large size flask at 200 rpm speed we can multiply our bacteria. Then after multiplication, we have to check the quality of broth. It should not have any contamination. It should be having sufficient cell load, around 10⁹ cells per ml. Then after this inoculum is mixed with the carriers. After mixing, we have to cure it, then quality check, then seal it into the polythene bags, and then store at the 4 to 6 degrees centigrade for their better shelf life. So this is the flow chart in the biofertilizer production. These are some of the pictures of root nodules, how root nodules looks alike for different crops. Like in this picture, we can see this sesbania. It is having the stem nodules also. White clover, mung bean nodules. This is chickpea nodules. So we can see here different type of root nodules found by the rhizobiums. So when we go for the isolation, we have to separate these root nodules and we can crush them. Then we have to inoculate with this nodule extract onto the suitable media plate so that we can get the rhizobium culture also. If anybody is interested in this procedure, I will explain because it is quite lengthy. Then there are inoculation technique which we will discuss into the detail. But why we inoculate? Some of the person will say that these bacteria are already present into the soil. So what is the purpose of adding extra? So placing very high population of the desired bacterium at optimum position for rapid colonization so that our target function can be achieved. That's why we apply biofertilizers to the soil. Then there are three main methods: seed inoculation, seedling inoculation, and soil inoculation. So among the seed inoculation, seed treatment is the most common method, in which we take 10% sticker solution. It can be prepared either by using jaggery or guar gum. So one packet of inoculant, which is of 200 gram, can be mixed with 200 ml of sticker solution. 10% jaggery we have to add, like for 200 ml, we need to add 20 gram of jaggery. Then after boiling of the sticker solution, we have to cool it, then mix our inoculum, and then after thorough mixing with the inoculum, we have to pour this slurry onto the seed, and one packet of inoculum is sufficient for the 1 acre of the seed. So after mixing with the seed, we have to shade dry it for 30 minutes, and then these shade dried seeds should be sown within 24 hours. So this is the flow chart of the seed treatment. Like, first of all, we prepare a sticker solution. After this, we cool it. Then we add our culture, then mix it. Then after mixing, we pour this slurry onto the seed. And after this, we have to shade dry it. Then our seeds are ready for sowing. If you will come in a physical mode, we will demonstrate it practically. Then seedling root dip. It is nothing but it is used for the transplantation crops. In this, we add one packet of inoculum to the 5 to 10 liters of water, and then we put our seedlings root for 30 minutes, and then immediately after that we transplant those seedlings to the main field. During this period, the bacteria get attached to the root system. Then in the main field application, we can mix our biofertilizer packets with cow dung or FYM along with the rock phosphate, so that we can get some better results. And in that mixture, we need to maintain this moisture content also. 50% moisture need to be maintained. Then it can be simply broadcasted during the land leveling or of the soil. Then for the quality regulation, some of these standards have been brought out to the market, and these are the Indian standard and international chemical standards for the biofertilizers. These standards prescribe quality requirements of inoculants in terms of the number of viable cells into the carrier material, stipulated expiry period, freedom from the contamination with other microorganisms, etc. The standards also prescribe the test procedure for assessing the quality besides the sampling procedure. So instructions for packing and marking are also given to ensure the consumer's protection from the substandard quality products. These are the quality control parameters need to be maintained, like they should have minimum CFU count of 10⁻⁷ per gram of the carrier. They should not have any contamination up to 10⁵ dilution. When we go for the quality check, when we take this suspension of our biofertilizer formulation, it should have only the growth of the desired bacterium. It should not have any contamination. Their pH should be in the neutral range. They should hold the moisture up to 30 to 40%. And the particle size of carrier is also fixed. It should be in the range of 0.15 to 0.212 mm international standard C. So this was about the carrier-based biofertilizer formulations. Nowadays in the market, liquid biofertilizers are also available. They are also, procedure is also same, but just a carrier material, their preparation and mixing is avoided. So directly they are preparing into the fermenters and packaging into the bottles. Liquid biofertilizers are liquid formulations containing the dormant form of desired microorganisms and their nutrients, along with the substances that encourage the formation of resting spores or cysts for longer shelf life and tolerance to the adverse conditions. The dormant forms, on reaching to the soil, germinate and produce a fresh batch of active cells. So these cells grow and multiply by using the carbon source into the soil or from the root exudates. and they perform their function. Advantages of liquid biofertilizers over the carrier-based biofertilizers. They are having longer shelf life, 12 to 24 months, but it was only 6 months in case of the carrier-based biofertilizers. There are no chances of contamination because a fermenter is a closed system. We do not need separate carrier material preparation, then mixing. So this procedure actually expose for the contamination. So there are the chances, but in case of liquid biofertilizer, we simply pack it from the fermenters only. So there are no chance of the contamination and no loss of the properties of our biofertilizer due to storage up to the 45 degrees centigrade. But in case of liquid biofertilizer, we need to store them at the refrigerate low temperatures, 4 to 6 degrees centigrades in the cooler environments. They have greater potential to fight with the native populations as we have added cell stabilizers are also there, nutrients are also there. And high population can be maintained into the liquid medium. We can maintain cell load more than 10 raised to power 9 cells per ml for up to 12 to 24 months. But it was only 10 raised to power 7 cells per gram of the carrier material. So it is almost 100 times higher. Then there is cost saving on the carrier material preparation, its pulverization, neutralization, sterilization, packing and transport also. Because carrier based biofertilizers takes larger volume. Because in spite of that 200 gram packet, only 50 ml bottle is sufficient. So we have reduced the volume by 1/4. So it saves on the transportation cost also. Quality control protocols are very easy and quick and very easy to maintain. They have better survival onto the seed also, into the soil also. Then there is no need of running the biofertilizer production units throughout the year because their shelf life is 12 to 24 months. So we need to run our biofertilizer unit once in a year. But in case of carrier-based formulation, where the shelf life is 6 months only, we have to run our biofertilizer units throughout the year. Its application is very easy by the farmers. They can simply mix into a tank or in a matka type structure, and they can simply apply with the irrigation water also. They can dip their seedlings, but it does not require very much skill. Though the protocols are same for the carrier based also, doses are 10 times lesser than the carrier based biofertilizers because their cell load is also very high. So we can use less amount of the dose liquid biofertilizer for the same area. So it has high export potential and very high enzyme activity since the contamination is very nil. So coming to the overall advantages of the biofertilizer, as they are eco-friendly and their application leads to the soil enrichment and the quality of the soil improves with the time. Though they do not show the immediate effects, but results shown over the time are spectacular. Like chemical fertilizers, when we apply the urea to the crop, on the next day it becomes lush green. But this is not the case with the biofertilizers. It takes the time and its effect lasts longer for longer periods. It have residual effects for the succeeding crops also. They harness the atmospheric nitrogen, make it directly available to the plant. They increase the availability of different nutrients, which we have already discussed. They improve root proliferation due to the release of the growth promoting hormones. Microorganisms convert the complex nutrients into the simpler forms so they may available to the plants. And biofertilizers contains microorganisms which promote the adequate supply of the nutrients to the host plant and ensure that their proper development of the growth and regulation of their physiology. They help in increasing the crop yield by 10 to 25%. They also protect the plant from various soil-borne diseases to a certain extent. In a study conducted with Azoospermillum brasiliensis in the grain yield of bajra, it was it was seen that with the application of azospirillum, we can see here with the 0 nitrogen dose, then there is 1/3 nitrogen dose, then half, and then full. We can see the effect in each setup. With the 0 nitrogen dose, without azospirillum application, the yield was 1208 kg per hectare. But with the application, it was increased up to around 200 kg. It becomes 1396 kg per hectare. In the same way, if we go to the full nitrogen application without and with application of the azospirillum, we can see here that increase in the yield also. So effect of cell load was also studied. Like if a bacterial formulation is having 10⁷ cells per gram carrier, its grain yield is 1.29 ton per hectare. But when the cell load is increased from 10⁷ to 10⁹, the yield of soybean was increased from 1.29 to 2.26, which is almost double. So they have a good effect on the yield. So, but though there are several constraints also, because as we know that biofertilizers are not much popular. So what are those constraints? We should know that the technological constraints like unavailability of good quality carrier material, then lack of qualified technical personnel into the production units, then the infrastructural constraints like lack of essential equipments, power supply, etc. Then financial constraints like non-availability of sufficient funds or problem in getting bank loans, because everybody is not having cash in hands to start a unit, biofertilizer production unit, which requires around 50 to 60 lakh rupees for initial startup. Then environmental constraints like seasonal demand for biofertilizers, simultaneous cropping operations, and short span of sowing / planting, so that farmer doesn't get time for such kind of applications. Then human resources constraints and quality constraints like lack of technically qualified staff in the production units, lack of suitable training on the production techniques, then unawareness of the farmers about this technology is the main problem in the adoption of the technology, because farmers are not aware of the benefits of the biofertilizers. We have to demonstrate them visually how we can apply them. So then marketing constraints are also there, like non-availability of right inoculant at right time, right place. There may be some other constraints like availability of a good quality strain, and availability of good quality strain at a particular location is also very important. Suppose a bacterium is isolated from Rajasthan, and if we apply it into the cooler environment, it may not be working very efficiently. So location specific strain are also need to be developed for such kind of biofertilizers. So this is the first biofertilizer patent which was filed by the US during the 1896. It was nitrazine. And then there is a provision of subsidy also to start up the biofertilizer production unit, like subsidy up to rupees 40 lakhs or 25% of the total cost, whichever is smaller. If your cost is more than 2 crores, then you will get only 40 lakhs. Or if your cost is around 1 crore, then you will get 25 lakhs. So whichever is smaller, it will be provided by the NABARD or National Cooperative Development Corporation. So these are some of the packets prepared by the our premier Indian Agriculture Research Institute. So farmers' awareness about the biofertilizer is very important. And some of the persons they ask that whether you should use a single strain or multi-strain. So there are different aspects regarding the single strain or multi-strains. If our single strain is compatible and competitive enough, then it is good. Otherwise, use multi-strain so that they can any of one of these strain can give the better results, but they should be compatible and competitive. In that case, they should not be antagonistic to the each other. That also need to be confirmed first. Thank you. Now the presentation is open for discussion.